everyone. I am going to talk about the chemical oxygen demand. First, we will see the introduction. COD is used to estimate the amount of oxygen that would be depleted from water sample due to the bacterial action under specific conditions of oxidizing agent, temperature, and time. In this test, there is the usage of strong chemical oxidizing agents such as potassium dichromate or potassium permanganate to oxidize the organic material in the sample of wastewater under conditions of heat and strong acid. Also, in this test, there is no interference from toxic materials. The test can be completed in 2 to 3 hours. COD is often used in conjunction with the BOD test to estimate the amount of non-biodegradable organic material present in the wastewater, and it is expressed in mass of oxygen consumed over volume of solution, that is milligrams per liter. COD is used in quantifying the amount of oxidizable pollutants found in surface water like ponds, lakes, rivers, and wastewater. And it is useful in terms of water quality by providing a metric to determine the effect of an effluent on the receiving water body. It measures all organic contaminants that are not biodegradable. Unlike for the BOD test, the toxic compounds like heavy metals and cyanides, which is present in the sample to be analyzed, do not have an effect on the oxidants used in the COD test. COD test can be used to measure the strength of the waste that are too toxic for BOD test. COD test is used to monitor the efficiency of the waste treatment plant. And now let's see the principle behind the COD test. COD is measured using a strong oxidants like either potassium dichromate or potassium permanganate under acidic conditions. A known excess amount of this oxidant has to be added to the sample. Once oxidation is complete, the concentration of organics in the sample is calculated by measuring the amount of oxidant remaining in the solution. It is done by titration using an indicator solution. COD is expressed in mg per liter, which is indicated as the mass of oxygen consumed per liter of the solution. And now let's see the materials that is required for carrying out this experiment. First, the water sample. You can take any water sample, either pond, lakes, rivers, or any other thing. And we need the potassium dichromate solution, which is having 0.1 normality. Then sodium sulfate solution, potassium iodide solution, then starch indicator, that is 1%, then water bath is required, then titration apparatus, and 100 ml conical flasks. And now let's see the procedure for the COD. First, the water sample is collected from any source, either pond or any other water body. 10 ml of water sample has to be poured in 300 ml conical flasks labeled as test 1, test 2 and test 3. Simultaneously, the distilled water is taken in 300 ml conical flasks labeled as blank 1, blank 2 and blank 3. Then 5 ml of potassium dichromate solution has to be added in each of the 6 conical flasks, and keep the flask in the water bath at 100 degrees C, that is boiling temperature, for 1 hour. Then the samples have to be cooled for 10 minutes. Then 5 ml of potassium iodide has to be added in each flask. Then 10 ml of sulfuric acid has to be added in each flask. Then titrate the contents of each flask with 0.1 normal sodium thiosulfate until the blue color disappears completely. And next we'll see the calculation for this COD. COD is calculated as A minus B into C into 8000 divided by the volume of sample in ml, where the A represented the titrant used for sample, that is expressed in ml, and B is the titrant used for blank sample, that is also expressed in ml, and C is the normality of the sodium thiosulfate, and the results will be expressed in mg per liter. Thank you.